Done, sir.
I'm very happy to welcome Dr. Ruchi Gulati Marwa today, and she'll be speaking on applications of biofilm. So, Ruchika, you can please take over.
Thank you very much, sir. At the outset, I would like to thank Dr. Singh and Professor Deshmukh for giving me this opportunity to speak on Microbiology Society Forum. And I feel that this initiative of Microbiology Society is having an immense contribution to this dissemination of microbiology and science in general. Today, I'm going to be speaking on a novel strain of Bacillus paralecheniformis that forms robust and resilient biofilms in cultured conditions in the laboratory. I'm going to be, talking about the structural and biochemical aspects of the biofilm produced by this organism, and also explore the spectrum of its potential biotechnological applications for the industry. So before I move ahead, I'm going to be speaking on and giving you a small introduction about what biofilms are, though many of you would be aware of this term. So biofilms are as ubiquitous as microorganisms themselves. So when we talk about the microorganisms, I'm referring to planktonic microorganisms when the microorganisms are existing as single cells. So biofilms are structured communities of tightly associated microorganisms, including bacteria and other microorganisms like fungi and algae, that are encased in a self-producing a matrix, which is called as an extracellular matrix, which is composed of extracellular polymeric substances and shortly called as EPS. Now, this matrix holds these cells together and effectively glues them to a biotic or abiotic surface. Now, the term biofilm was coined by a great scientist, Bill Costerton. I'm going to be coming to the work of this scientist a bit later in 1985. But the concept of biofilm is not something which is very new. In fact, it is very, very old. And the description of the biofilm dates back to the 17th century when Anton von Leeuwenhoek saw microbial aggregates, now known as biofilms, on the plaque from his own teeth. Now, the extracellular polymeric matrix of the biofilms is a polymeric jumble of proteins, polysaccharides, nucleic acids, essentially DNA, water, ions, lipids, etc. And these biofilms can form under both natural and artificial conditions. So, for example, in the laboratory, in cultured conditions, biofilms can form both on agar surface and in submerged or floating forms in liquid culture medium. So this communal lifestyle of the biofilms is very different from the planktonic life. And it is a highly regulated and coordinated series of events that take place involving some environmental triggers, quorum sensing that involves the synthesis of certain molecules like autoinducers, and other small signaling molecules to give rise to this emergent form of life. So biofilms have a great importance for public health because they can create impossible to eradicate infections on central lines and catheters. For example, they can form at various places inside the human body in the case of infections. And they can also have an industrial impact because they can foul everything from sewer lines, water distribution systems to our teeth. And thus, they have a grave and negative aspect and impact on the public health. Uh, with regard to their persistence, in the biofilms, the pathogens have a high degree of persistence, and therefore they can cause some very, very difficult to clear infections. In 2002, as I mentioned earlier, Costerton was the person who had used and coined the term biofilms. So in 2002, after about 10 to 15 years of his research, one of his collaborators, Donlan and Costerton, then they offered the most salient definition for a biofilm. And they stated that biofilm is a microbial-derived cell community characterized by cells that are irreversibly attached to a substratum or interface, or to each other, and they're embedded in a matrix of extracellular polymeric substances that they have themselves produced. And in this community, they exhibit an altered phenotype with respect to growth rate and gene transcription. So, which means a large number of biochemical changes, phenotypic changes, and transcriptional and translational events are taking place during the process of biofilm formation. And if you can see and look at these pics here, you can see that at what sites in the human body, for example, they can form biofilms right from as simple as a contact lens, or on the lungs of a cystic fibrosis patient, on a catheter, etc. They can also cause a microbial induced iron corrosion, leading to the structural damage in the industries, and they can also spoil the structure of the ships floating in the water. So therefore, biofilms have a very negative impact on the public health. But this particular talk today is going to be focused more on their positive and beneficial aspects rather than on the negative impact. So now, when the biofilms are formed, it goes through different stages in the life cycle of the biofilm forming bacteria. So if the planktonic bacteria, depending on the environmental circumstances they are subject to, and they find a suitable substrate to attach to, they will attach to that surface and then form a monolayer of cells by the production of certain matrix producing cells, which will eventually produce the extracellular matrix of the biofilm. And then they form a microcolony. So till this particular first or two or three steps, this process is reversible. But once this microcolony and a thick layer of these cells form in a multi-layer, the process becomes irreversible, and then a mature biofilm is formed with a characteristic mushroom-shaped fruiting body, which is consisting of extracellular polymeric substances. Now, the important point to remember here is that the dense aggregation of cells in a biofilm can lead to a lot of heterogeneity with respect to environment inside the biofilm and also with respect to the cellular phenotypic characteristics of the cell. So, for example, the nutrient gradations which are formed inside a biofilm because of its thickness can trigger cells to differentiate into different phenotypic states. And as a result, these cells can form subpopulation of cells within a single biofilm and to adapt to the physiological conditions of that particular biofilm. And they can also differentiate functionally. They can become functionally distinct and then lead to a division of labor and a large number of cooperative interactions between the cells, and as a result of which there is a lot of functional and phenotypic heterogeneity within a biofilm. So they work as a community and have an interaction with each other on one-to-one basis in terms of signaling and in terms of providing nutrition and other growth factors to the cells of this community. Now, when the biofilm community matures, then the cells might detach and they can revert to the planktonic growth and then start a new cycle for the biofilm formation. Now, the EPS of biofilm is a very dynamic component of the biofilm that contributes to their structural complexity. And it is more than a physical scaffold or a structural support for the biofilm. So there was a conference from the American Society of Microbiology in 2007 on biofilms in Quebec, Canada. And there it was stated that biofilms can be metaphorically called a city of microbes. The EPS represents the house of the biofilm cells, where these cells of the biofilms would essentially reside. So the EPS of the biofilms offers a large number of advantages for the cells of the biofilm. It can improve the biofilms community's ability to scavenge both water and nutrients from a nutrient deficient environment. It can protect the microorganisms in the community against stress or damage from exogenous contaminants like antibiotics and pollutants or toxins, etc. It is also an ideal place for exchanging genetic material and maintaining a large and accessible gene pool. So, therefore, even the antibiotic resistant genes can be transferred to other microorganisms in this community through horizontal gene transfer. It also provides a functional and structural support for the biofilms, making available various other nutrients from the environment and providing a physicochemical heterogeneity for the support of the biofilm. So, which means that EPS is a very important component of the biofilms, and biofilms can be formed both as monospecies biofilms or multi-species biofilms, where different kinds of microorganisms can be part of this community, and thus it can lead to a large number of interactions, both competitive and cooperative interactions in the formation of the biofilms in the natural environment. So coming back to this great scientist, Bill Costerton, he was the one who had actually started the pioneering work on biofilms in the around 1985. And he was a very passionate scientist and was involved in directly observing living complex microbial communities in all kinds of environment in nature, for example, in rivers, on rocks, in the health system, in the streams, on the trees, etc. And wherever he could find the bacteria or other microorganisms growing as biofilms, he was very keen to observe them, under these conditions. And he was a very charming personality for all his students, his colleagues, and he was able to disseminate his enthusiasm of science through them and nourish their scientific temperament. He was author of more than 600 peer-reviewed publications, a prolific writer, and also, an excellent public communicator. And he was able to communicate the concept of biofilm formation right from a lay person to a medical professional like a doctor. So he is regarded truly as the founding father of the field of biofilms, and I believe that his name should be in every textbook because he gave a new dimension to the field of microbial ecology in general. I believe, and this particular talk is, a small tribute to his immense contributions to the field of biofilms. He started his work in the field of biofilms in the University of Calgary, Canada, and then he resigned from there and then joined as a director in 1993 at the Center for Biofilm Engineering in Montana State University. And this particular institute at MSU is helping the industry to find solutions to the biofilm challenges, including public health impact, and also developing biofilm uses for biofilms in for a number of industries. So therefore, biofilms also have a lot of potential biotechnological applications, which I'm going to be talking about in a bit when I talk about my own research and our group's endeavor in this direction. Uh, so then, this Costerton Biofilm Center was opened in University of Copenhagen to honor his immense contributions to the research. So as they rightly say that biofilms are about 3.5 billion years old, but biofilm research is only 35 years old. So there is a lot of potential in this area of biofilms to carry research on. So, as I mentioned earlier, the EPS of biofilm is contributing to the functional as well as structural integrity of the biofilms, and also towards the architectural beauty of the biofilms. Biofilms can adopt a three-dimensional shape due to the presence of their EPS, and this EPS composition can vary from one microorganism to another, or from one bacteria to another. And generally, among gram-positive bacteria, when we work on biofilms, we use Bacillus subtilis as a model system. And in the gram-negative bacteria, we use Pseudomonas aeruginosa as the model system for studying biofilms. So as you can see in these photographs, this is a biofilm formed by the Pseudomonas and the Bacillus subtilis. And the EPS components of the biofilms are responsible for the wrinkled colony appearance of these biofilms, a highly mucoid appearance of the biofilms, and also the fibrillar network appearance of the pellicle. And they're also responsible for the overall resilience of the biofilms, which make them highly resistant to antibiotics and other chemical components. Let us now look at this small video that is a time-lapse video for the formation of a multi-species biofilm of Escherichia coli and Acinetobacter bailii.
Hello, Dr. Ruchi.
Sir?
Please go ahead.
Yeah, I'm just finishing this video. Yeah.
Okay. Very nice.
Right. So as you can see, that the architectural complexity of the biofilms is immense and highly varied from one organism to another, and under different environmental conditions or culture conditions in the laboratory, their phenotypic characteristics can also change, as we can see in this particular slide. So now the architectural and structural complexity of the biofilms is a subject of great study among the scientists, and they're also trying to understand the genetic and molecular basis of biofilm formation that will help them to understand both the how to tackle the negative aspects of the biofilms and also to use them for their own benefit. Scientists are now using computer simulations and mathematical modeling to understand the architectural complexity of the biofilms. Now, a lot of research is now being focused on the beneficial aspects of this biofilm, as we can see from this publication of 2016. But traditionally, biofilms have been known to be used a lot in wastewater treatment. They are inherently formed in wastewater treatment for the treatment of waste, and now we are building up wastewater bioreactors where biofilms can play a very important role in effective wastewater treatment. Biofilms can also form in the soil and can promote plant growth. Apart from that, they can have a large number of biotechnological implications. So, the research on biofilms in our Department of Microbiology at BCAS started in 2018. And we have considered this particular research project as a pedagogical case study. So I'm going to be talking about that how we are trying to understand the development of the biofilm from a novel strain of Bacillus species, and how this particular project has transformed into a pedagogical case study for us, because we have tried to involve our students in this research endeavor at the undergraduate level, and that has helped us improve their profile and make them ready for their future destination or higher education. So when did this, research on biofilms begin for us? It was in 2018 that we isolated this peculiar and very beautiful looking bacterial colony that was on a nutrient agar plate that had been exposed to the indoor environment of the chemistry laboratory of our college. It was a routine experiment for our students for exposing their plates to isolate the microaeroflora from the environment. But this we were intrigued towards this particular beautiful looking colony that was a flower shaped colony, a spreading colony with a core region that was dry and wrinkled, but it had a peripheral region that was highly slimy and mucoid. And this was that caught our attention, and we wanted to understand if it was a single colony or or a group of two kinds of bacteria growing together. So we went on ahead to purify this particular colony by pit plating, and then we did some detailed growth studies and phylogenetic studies on this particular bacteria, and also characterize the EPS from this colony to establish it as a biofilm producing bacteria. So, as I mentioned, that this particular bacteria grew as a segmented colony. So, we wanted to understand if this particular kind of a phenotypic character was, recurrent for this particular organism. And we purified this particular bacteria. And every time we subcultured it, it grew as a flower shaped colony with a distinct spreading mucoid region and a creamy wrinkled core center. We did some basic microbiological tests on this colony. It was a bacteria which was gram positive with subterminal endospores, a thick capsule, a very, very thick capsule, in fact, because of the high quantity of mucoid production during the growth of this particular organism. We did a series of biochemical tests and eventually identified this organism as a Bacillus species. When we grew this organism in a nutrient broth medium in liquid culture conditions, it formed a very thick biofilm pellicle at the air liquid interface. And by the well known crystal violet assay, we were able to establish it as a colony biofilm and a pellicular biofilm in laboratory culture conditions in the normal nutrient agar and nutrient broth medium. Then we studied the growth cycle of this organism, and we found that in the broth conditions, we found that initially the cells are planktonic in nature, and then they start aggregating due to the production of the extracellular polymeric substances in abundant amounts, and then they become chains. and then a very thick, mature biofilm is formed, which is typical of any biofilm bacteria. And the important point to remember, to remember here was that a well-developed biofilm pellicle is formed within 16 to 18 hours of incubation. So therefore, it was a very fast and a rapidly producing biofilm. We did, large number of culture studies on this organism by manipulating the culture conditions, because from the literature we came to know that in the laboratory, manganese and glycerol are some of the triggering, factors for the production of a more robust biofilm. And we found that when we supplemented our nutrient broth or LB medium with glycerol and manganese, we had a more robust biofilm formation and a more three-dimensional biofilm formation in comparison to the medium without these enhancers. So as you can see, that in medium one, which is only nutrient broth or urea broth, we have a thin biofilm formation, though resilient, but a very thin biofilm formation. However, in medium 4, supplemented with both glycerol and manganese, we have an architecturally complex three-dimensional biofilm with a very thick phenotypic character. So it was a very robust biofilm. Our students, recorded a time lapse video using the hanging drop method, which we normally use to check the bacterial motility in the laboratory, to understand how these cells are aggregating to form the biofilm. So initially the cells are planktonic, but then they keep on attaching and forming larger aggregates of the cells to form the biofilm at the air liquid interface in a water droplet. So these aggregates are increasing and increasing in size, and the planktonic cells are now decreasing in number in the final biofilm formation. We also did some SEM studies to understand and compare the biofilm formation in all these 4 different media, supplemented with glycerol and manganese, and also both of these components together. And we found that, as you can see, that such a beautiful architectural complex scaffold of biofilm is formed in the LBGM medium. Also, when we compared the quantity of the biofilm of this organism with other organisms, we found that it produces the biofilm in much, much larger quantities in comparison to Pseudomonas aeruginosa and Bacillus subtilis, that is used as the model systems for this particular biofilm formation. So therefore, it was important for us to investigate further into the characteristics of this biofilm. And before we did that, we wanted to identify this organism. And so we did some 16S ribosomal RNA sequencing and whole genome sequencing by this organism. And we identified it as Bacillus paralecheniformis, named after our college in 2018. And the biofilm colonies of this particular strain are very, very phenotypically different from related strains, Bacillus lecheniformis and other Bacillus species, for example, Bacillus subtilis. We published the whole genome sequence of this organism in Microbiology Resource Announcement of ASM in 2021. We further went ahead to understand the resilience of the structure of this particular biofilm by this organism, and we did some organic solvent tolerance and viability studies. And this biofilm was highly tolerant and stable in various organic solvents. And even after treatment for 2 hours to 4 hours, the cells of the biofilm were very viable post treatment, as can be seen from these two graphs. We also did, by the water droplet method, the hydrophobicity of this biofilm, and we found it to be very hydrophobic. As you can see in the medium, when the colony is grown on LBGM medium, it forms a complete round drop with a very high contact angle, thereby, proving that this particular biofilm is highly, highly hydrophobic. We also did some antimicrobial susceptibility studies, and we found a very unique phenomenon happening in this biofilm, and that was that the extracellular polymeric substances, or the mucoid material that this particular strain produces in abundant amounts, can make a bacterium susceptible to antibiotic initially to a temporally resistant bacterium, especially in the case of chloramphenicol and erythromycin, within 24 hours. And the quantity of EPS was enhanced considerably in the presence of these antibiotics, which means that due to the stress present in the environment of the bacteria, the EPS production is significantly enhanced on these medium. We also, wanted to characterize the EPS of this biofilm, and we did some FTIR, UV visible spectrophotometry, NMR studies, and confocal microscopy studies. And two main components of the EPS were characterized. One of these components was a secretory component, which is a polymer known as gamma polyglutamic acid, which is an industrially valuable biopolymer. And the second was a proteinaceous component, another proteinaceous component, which is called as amyloid fiber. This was seen by the thioflavin T staining performed during the confocal microscopy. So as a result, we were able to characterize these two components, and polygamma glutamic acid is responsible for the initial formation or initiation of the biofilm and overall robustness of the biofilm, as we can see and from the literature available to us. And the amyloid or the protein fibers were confirmed through confocal microscopy, and they are responsible for the hydrophobicity of the biofilm and the fibrous appearance of the pellicle and wrinkled appearance of the colony biofilm core. So coming to this, polygamma glutamic acid, an important secreted polymer of this biofilm, it is an industrially important biopolymer of glutamic acid with various biotechnological applications. It is a non-toxic polymer, biodegradable, water-soluble, and edible, and therefore presents itself as an important candidate for a production from this organism and can be used for various applications. So now, after looking at the structural and biochemical aspects of biofilm formation of this novel strain, we wanted to explore the biotechnological prospects of this isolate through its biofilm. And the first was the potential role of gamma PGA in various applications. And as you can see, gamma PGA is an inert polymer. It can be used in medicine, in wastewater treatment, in agriculture, food, cosmetics, etc., and therefore, has a polymer of diverse applications. So therefore, if we are able to engineer this organism or Bacillus parallacheniformis for a large scale production of this biopolymer, it would be very valuable from the industrial viewpoint. We are also looking at another aspect of the biotechnology of this particular biofilm, and that is bioremediation. So we are carrying out biofilm-mediated bioabsorption of toxic dyes, and in the first phase of this research, we have tested 4 dyes: malachite green, crystal violet, Congo red, and methylene blue. And we have been able to see that this particular biofilm is able to absorb more than 90% of all these 4 dyes, and even under different varying or wide range of pH. So as we can see, that the entire medium in which this particular dye was dispersed, the biofilm has taken up all this dye, and this is a very important indication of this fact that we can use this particular biofilm for bioremediation purposes, for the removal of pollutants like toxic dyes, which are very widely used in the textile industry. So now the fundamental question is that we need to, if we want to understand the biotechnological applications of this biofilm, we need to regulate the PGA production. We have to engineer the medium in such a way that that we are able to divert the growth of this bacteria towards PGA production and less towards biofilm formation. So we have to understand what are the medium and inducers required that can improve and optimize the production of the PGA. We have a very stark difference in the phenotype of this biofilm from other known biofilm producers. So it can be used as a model system for biofilm research. And if we are able to, answer some of the molecular reasons behind the biofilm formation or the genetic reasons behind the biofilm formation, we'll be able to control the biofilm formation and explore it further for various biotechnological applications involving studies like metabolic engineering, mutation studies, structural biology, biofilm engineering, etc. Now, another important aspect of PGA production was that though initially PGA was part of the biofilm formation in the biofilm growth cycle of this organism, it was not eventually the part of the mature biofilm pellicle. And it was secreted completely into the liquid medium. So, therefore, it can be easily isolated for industrial purposes from this broth. These days, a new field of biofilm research has opened up, and this is called as biofilm engineering, which uses synthetic biology and genetic engineering techniques to reprogram the biofilms to improve their functions and enhance the production of value-added products. Like, for example, if we can attach, or we can program the biofilms to produce certain industrially valuable enzymes like amylases, lipases, proteases, etc., they can act as microbial cell factories for us to produce these value added products in large scale, and also we can reuse these biofilms for continuous production of these value added products in industrial fermenters or bioreactors. Similarly, biofilms can also be, reprogrammed or, manipulated to do waste degradation on a very large scale, and so they can help in removal of pollutants, different kinds of pollutants, including hydrocarbons and other toxic dyes, heavy metals, etc. Apart from that, biofilms can also be, produced on a renewable scale using 3D printing, and then they can be converted into living materials that can help for various industrial purposes, like for example, if we have a bone replacement, and we have some gaps in our joints, so these biofilms can act as living functional materials to fill those gaps and help the patient in the recovery of the bones and the joints. So therefore, they have a large number of applications. Similarly, biofilm produced through 3D printing can also be used in the construction of the buildings, in the building materials, as renewable materials for the robust structure of the buildings. So we have a large number of applications through biofilm engineering. So this was, the main research that we have carried out on this particular novel strain. And most of the studies on the Bacillus biofilms are restricted to Bacillus subtilis. But we, propose that because of the high phenotypic variation of this isolate from the other known biofilm producers, this organism is an interesting candidate to be used as a model system to understand the molecular mechanisms underlying biofilm formation. Medium engineering can help us to enhance the synthesis of polyglutamic acid from this particular organism. And we have also got some promising results with respect to bioremediation of the toxic dyes polluting the environment, and which we can scale up for extrapolating it to textile dye effluents. Now, coming to the second, perspective of this research, we started this research work in 2018, and at that particular time, three of our students joined us in this minor project that was supported partially by the Star College Scheme, Department of Biotechnology, Government of India. And in the course of the progression of this project, we found that it was a very fruitful case study for us by using it as a pedagogical tool to enhance student research outcome, giving them an experience of a lifetime to work in the field of microbiology research in their own laboratory, and understand how understand the nuances of research and motivate them towards higher education through this initiative. So if we look at the advantages of for any undergrad student or any postgraduate student of having a research experience before they go for PhD, etc., it is a very rewarding experience for them, because they can acquire some basic and advanced skills through these research projects, giving them a very suitable hands-on experience for doing their further research in their journey towards PhD. And also, if they want to work in an industry, this experience can come in very handy for them. Through this research, they will read more, and we all know that reading is rewarding. They will collect research papers, they will review literature, they will update their knowledge about a particular topic. And as I mentioned, they will acquire new skills, they will publish their work. We have published one paper, we're in the process of publishing more papers, presenting our work in the conferences. They will know how to do citations for the references, use reference managers like EndNote, Mendeley. They will be having a clear idea about their career direction, whether they want to go in for research or for some other field of study. They'll be able to face interviews with more confidence and acquire communication and writing skills. They will also work as a team and collaborate. And also, to add a little extra salt and pepper to this, they will improve their CV and profile and help them think out of the box. What I believe is that microbiology has never failed to surprise me, and it has never bored me. So I think it is a very interesting field of research, and this forum of microbial society is doing the right work to disseminate the science of microbiology to each and every person involved in this particular society. This work has, brought us 4 poster awards through various international and national conferences, and all thanks to our students who have worked untiringly, for this particular project since 2018. And this is our research group and collaborators. Professor J.S. Virdi and Professor Rani Gupta are my teachers from Department of Microbiology, University of Delhi, who were the first one to motivate us to continue this particular project. Dr. Deepti Jain is my research collaborator from Regional Center for Biotechnology, Faridabad, and she was the one who was responsible for the whole genome sequencing of this organism. My colleagues, Dr. Purnima, Dr. Rityakshi, and Dr. Neeru from my department at the college. We have trained these Pooja Gupta, Anirban, and Tanu were the first 3 students who had initiated this project, and then were joined by these students in the phase 2 of the project: Hitik, Kritika, Kaushik, Abhiman, Priya, and Jyotsna and Pragya. And then these are the students, Shashwat and other students who are um, doing this project at the phase 3 right now. Right now, I'm in the UK on leave, but I'm still handling this project remotely with the help of my colleagues at the department. And we have initiated a research collaboration with Professor Andrew McBain at University of Manchester, UK, to further understand the biofilm formation from this organism. And we are also collaborating in another perspective of biofilm research, and that is dry biofilm formation. So dry biofilms are a very cryptic phenomena that happens in the healthcare and food industries and other industries, and it is a very interesting and novel field of research for which we are collaborating with Professor Andrew at University of Manchester. So this was all about my research work in the area of biofilms, and I would like to thank each one of my collaborators, my colleagues, my students for supporting me in this endeavor. And this study has been partially funded by the Star College Scheme, DBT Government of India, and by Dr. Deepti Jain's Laboratory Regional Center for Biotechnology, Faridabad, Haryana. So thank you very much, and I'd be very happy to take any questions in this regard in this particular field of research. Thank you very much.
Thank you very much, Dr. Ruchi. That was an excellent talk by you. I really enjoyed the talk and came to know new developments in biofilm. I want to ask you a couple of questions.
Yes, sir. Thank you.
Can we induce biofilm formation in bacteria that do not form biofilms?
So now, interestingly, this question has a very unique answer. When Costerton started working in the field of biofilms and started understanding how these are formed in the natural environments, like in the rivers, streams, trees, etc., he found that biofilm growth is a default mode of life for bacteria and other microorganisms. So, for example, it is something which is the natural tendency of the microorganisms to form biofilms in the natural environment. So planktonic mode is just a chance mode of growth for these organisms. So mostly when microorganisms live in the natural environment, for example, in the soil or on the plants or on the rivers or on the rocks, they form biofilms. And so which means that genetically, all microorganisms will have some gene or the other to form the biofilms under natural conditions. However, under cultured conditions in the laboratory, different microorganisms might have different culture conditions to form the biofilms, but in cultured conditions, not all microorganisms will be able to form the biofilm, because we will not be able to understand specifically what specific culture conditions are required for a biofilm to form in a laboratory. But under natural conditions, as advocated by Costerton, biofilms are formed by almost all microorganisms, and it is a phenomenon that has been happening since 3.5 billion years ago. So therefore, it is something which is happening, but we came to know only about this particular phenomena after Costerton gave a new dimension to the field of biofilms and the field of microbial ecology.
Okay, thank you so much for the good answer. Is a consortium of bugs better than a single microbe?
So can you repeat the question, please?
Is a consortium of bugs better than a single microbe in forming biofilms?
See, it depends upon where these biofilms are being formed. So, for example, if we want to apply these biofilms in wastewater treatment, so I would prefer a multi-species biofilm having a consortium of different microorganisms to form the biofilm, because then these microorganisms will have a diverse range of degradative abilities, because of which they can degrade a large variety of waste matter in the waste waters. However, if we are doing a specific purpose, a specific application for these in the industry, then in those cases, we can have a monospecies biofilm, because we are directing our application towards a particular action of these biofilms. So therefore, when we talk about wastewater treatment, we should have a multi-species consortium of these biofilm bacteria or other microorganisms that will help in a more effective treatment of the wastewater.
Yes, yes, because wastewater is having a variety of pollutants. It may have
Yes,
hydrocarbons, it may have pesticides, it may have
exactly.
heavy metals, and so on, right?
Yes,
So, consortium is definitely better in cleaning the environment.
exactly. Yeah.
Yes. If anybody has a question, please ask. I think in the chat box there are some questions. In the chat box.
I will look at the chat box now.
Please, please.
Okay, so I will first, Dr. Prasoon has asked this question: what governs the hydrophobicity of the biofilm? Essentially, as I mentioned, that in our case, for example, amyloid fibers were the major component of the EPS. So anything which is related to high polymeric components like proteins will add to the hydrophobicity of the biofilms. And also biofilms also have lipids. So we have not yet characterized our biofilm from the lipid perspective, but we know that the amyloid fibers are the major component of the biofilm that is attributing to the hydrophobicity of the biofilm. The second question is, can we get the protocol for time-lapse biofilm formation? So it is a hanging drop method where we are, fitting a camera to the top of the trinocular microscope, and we then record the movement of the bacteria in the hanging drop using a cavity slide to record the video of the biofilm under the microscope. So this is how our students had recorded this video using a microscope attached to the using a camera attached to the microscope. Yes. So am I able to answer your questions, Dr. Prasoon?
Yes, ma'am.
Yeah. So third question is, please elaborate more about dry biofilms, how they get formed. So as I mentioned, dry biofilms is a very cryptic phenomenon. And so technically, if a bacteria, say, for example, in a healthcare system, we are using endoscopes, right, for the patient. So in the endoscope, we use them, and then we clean them and then put them for drying out, and then we reuse them. this happens generally in India because we have a very compromised healthcare system. So we cannot have new endoscopes every time. So we reuse these endoscopes. Okay. So when we hang them for drying, some of the bacteria remain. So these bacteria have already are able to form the biofilms in the endoscopes because of the fluids of from the patient that have gone into these endoscopes. So they get all the nutrients from the patient's fluids, from the body fluids of the patient, like blood and other internal fluids. So as a result of that, when these endoscopes are drying, these biofilms also dry. And when they dry, it is difficult to see them also, and also difficult to remove them because they are resistant to the cleaning procedures of the endoscopes. For example, we are using some chemicals to remove these dry biofilms or these biofilm bacteria. So when they become dry, they become all the more thinner and they become invisible almost. So it is a very cryptic phenomena. So therefore, we don't know at the moment that what is causing these biofilms to dry, and is EPS still a part of these biofilms or not? These are also some of the unanswered questions to these dry biofilms. So therefore, a lot of research is needed in these areas because these dry biofilms can create a lot of havoc for the healthcare industry. Because if by the routine cleaning procedures they are not removed, then they can transmit pathogenic pathogenic bacteria through these biofilms. For example, Staphylococcus aureus, Pseudomonas aeruginosa. All these are highly nosocomial pathogens found in hospitals. And if they form these dry biofilms, and they are forming these dry biofilms, we know that it will be difficult to remove them. So we have to find the reasons behind the formation of these biofilms and how they can be cleared from the healthcare systems and other industries for that matter, food industry, etc. So Dr. Prasoon, any other question?
No, no. Thank you, ma'am.
Okay.
Hello, madam.
Dr.
Yeah,
Bharti
we have one
asked you.
question.
Can I answer Dr. Bharti's question first? Dr. Bharti's question is, what's the difference between pellicle and content in mucoid colony? So pellicle is the complete biofilm. And within that pellicle only we will have the extracellular polymeric substances and the bacteria, if I've understood your question correctly. And
Yes,
the extracellular polymeric substances are the ones which are responsible for the mucoidy of these, or the mucoidy of these biofilms. But not all biofilms are mucoid. That is also another important. Like for example, Bacillus subtilis biofilms are generally the NCIB strain, which is the type strain used for biofilm formation, is not mucoid. The colony biofilms are not mucoid as what we have observed in our strain, because our strain had a segmented colony appearance. It had a core region that was dry, but the peripheral region that was mucoid. But in some other bacteria, we will have a lot of mucoidy as well. But that mucoidy may or may not be due to gamma PGA. It could be due to other polymeric substances as well. So every biofilm is different with respect to the EPS composition and the biochemical characteristics of this EPS, and also the structural framework of this of the colony or the pellicle. Or if they are forming a submerged biofilm, then definitely it's going to be different.
Mm-hmm.
Is there
Okay,
any
but
bacteria?
yeah,
Tell me. Yeah, please.
one more thing. And when we do the assay, there are many methods. Like the one, if we are getting a colony, I mean, in a broth, lot of pellicle is there, and if that only is a biofilm, can we just wash off the broth and stain this one and grade them
Yes.
as
Yes, we
high,
can
low,
do.
and moderate biofilm?
Yes, we can do that. That's
Huh.
what we have done. That's what
Hmm.
we have done when we have quantified the biofilm.
Hmm.
In the, am I
Like there are some methods. Hello.
am I
Hello.
audible to you?
Yes, madam. And there are
Okay.
some methods like in 96 well plate they do,
Plate.
no? So
Yes.
which is the more perfect one or nothing like that?
No, there is nothing like that. There is no perfect one. The
Okay.
one that works for you is the perfect
Hmm.
one. Like, for example,
Okay.
these days people have started growing biofilms on coupons, glass coupons and steel coupons, because they
Oh.
feel it gives them a more broader surface for the biofilm to grow, and
Hmm.
also help them to analyze the biofilm through confocal microscopy and other microscopic techniques
Hmm.
directly on the
On
coupon.
glass.
On glass coupons, and there are some round disc shaped coupons which are
Huh,
available.
huh, huh. Okay,
Okay,
okay.
on which
Huh.
they grow these biofilms. So they put these coupons in the medium, in the broth
Hmm.
medium, and the biofilm grows on them. and then they can analyze them by crystal violet assay, or they can directly analyze them by staining under confocal microscopy.
Correct.
So there
Okay.
is
So any method.
no standard method. No standard method.
Okay. Thank you, madam. Thank you so much.
Thank you. Is there any bacteria that never forms biofilms? So Prasoon, are you a microbiologist by any chance? You are?
Yes, ma'am. Yes, ma'am.
So you would be knowing that out of all the known microorganisms on Earth, we only know about 1% of them, right?
Correct.
99% of them are not even culturable in the laboratory. So I do not have an answer to this question, because we cannot, unless and until we sequence all of them, culture all of them, will not be able to know that. But I can say that much, that most of the bacteria or other microorganisms, for example fungi, which are unicellular, would be able to form the biofilms, because they might be having the genes to do that. And it is kind of a default mode of growth for all these microorganisms in the natural environment. In the culture conditions, it would be difficult to answer, because then we will have to screen every bacteria to understand that.
Then while doing research, how can we keep like a kind of negative control that, okay, when we
Negative
culture
control would
this,
be the culture coming from a culture collection that is not forming a biofilm under cultured conditions. So for example, we have many Bacillus subtilis isolates or strains available in the American type culture collection or other culture collection centers, which do not form the biofilms under laboratory culture conditions, even in the presence of certain inducers, which means they might require some other inducers, or they are not able to form the biofilms under the culture conditions. So we cannot say that, but controls would be those which are planktonic essentially in the culture conditions in the laboratory.
okay, so how can we select such a culture like ATCC? Means you said that
We
there
have
are some.
ATCC cultures, and we will have, when we look at the research papers, we look at the research publications, people generally, so for example, Bacillus subtilis NCIB strain 3610, this strain doesn't form biofilms in the normal nutrient broth medium or normal nutrient agar. Okay, only when it is supplemented with glycerol and manganese, it forms the biofilms. So that is the control for this strain, without added manganese or glycerol, right? Our strain, luckily has, or fortunately, or maybe unfortunately, has the capacity, inherent capacity to form the biofilms, under both inducing and non-inducing conditions. That is, in the absence of the glycerol and manganese and in presence of glycerol and manganese. So which means that we will have to use a strain which is planktonic from the culture collection as a control.
Okay.
But the point is, when do we require a control? That will that will guide the choice of the control. If we require a control with respect to, say for example, organic solvent tolerance, then I would be using my own culture without organic solvent to act as a control. But if I require it for some other structural studies, or for comparison, or for an understanding the transcriptomics or genomics or proteomics of a planktonic culture in comparison to my biofilm culture, definitely I would then require a planktonic control.
Right. Yeah, this is a very interesting thing. So I think if there is no more question, then we can conclude the session.
Sir,
Dr.
I have a
Ruchi,
question. Sir, I have a question to ma'am.
we can take one question.
Yes.
Ma'am, is there a possibility of growing co-culture of a high biofilm producer and a low biofilm producer together and study if it can produce a high or moderate biofilm?
It depends. It will all depend upon if we are able to crack the culture conditions for them to grow together. I showed you a colony biofilm time-lapse video in the beginning with Acinetobacter, and so they were able to grow the colony biofilm together. But they must have given them some stress conditions for them to grow together, because for them it was a cooperative event of these two organisms growing together and then surviving in a colony biofilm. So definitely it is possibility. Multi-species biofilms can be grown in wastewater in bioreactors in the laboratory. It is known to us. So depending upon the two cultures coming together as a co-culture, growing them as a as a biofilm in which there are two indistinguishable entities will all depend upon the manipulation of those culture conditions. And it will depend upon the kind of bacteria that is forming those biofilms.
Okay.
Right.
Okay.
Right.
Thank you. Thank you.
Okay. Thank you very much, Dr. Ruchi, for the excellent talk you gave. And this will be on YouTube very soon. So many
Thank you
others
very
can
much.
listen to your good talk. Thank you
Thank
very
you very
much for
much,
the talk.
and for giving this opportunity. Thank you very much, sir.
Thank you very much.